Welcome, Thomas. I'm glad to have you here at the Max Imaging and Special Biology Days in your new role as a co-founder of Vibraint. Maybe let's start with that. Can you tell us a little bit more about the mission of your new company?
Yeah, absolutely. So first of all, thanks for having me here. We founded the company about a month ago now, Vibraint about a month ago now. And our general mission is to, I think, unlock neuroscience research across the third dimension. And the idea is to integrate both anatomical, structural, and molecular data within the same reference space. And I think the motivation started from our own frustration as neuroscientists, from having fragmented data sets that are really difficult to make sense of, especially in the brain that is such a complex organ to work with, to essentially streamline the pipelines using 3D imaging to really go as deep as we can into the problems we're interested in.
So why exactly should a biomedical or biopharma company be interested in 3D imaging at all? What are the clear advantages compared to more traditional workflows?
Yeah, so I think for pharma, ultimately what matters, right, is to understand how their drug works. Not only how it works, but also where it works. And, with traditional methods, usually you just get a small snapshot of what's happening, where the drug is distributed and how it acts. Here, I think for pharma, the main benefit is that with the technology that we have, they can assess how the drug distributes and works across the whole organ with cellular or even subcellular resolution. And so this has pretty important consequences because it's essentially better go, no go decision making, better maybe compound stratification, and just faster decision making for them.
Can you give us some numbers to get a feeling on how much time and work you can save by going for a 3D approach compared to something more traditional?
Yeah, that's maybe a little bit harder to estimate, but 2D is very tedious. It's human laborious. It probably takes a couple months of work of just human labor to slice tissue, image it, and I'm not even speaking about the analysis part. But in 3D, it's basically a fixed amount of time that you usually set for an experiment. It's much more scalable in a sense that you can run hundreds of samples, and within a month, essentially, you have your full analyzed, unbiasedly analyzed data set.
Yeah. When talking about 3D imaging, we must discuss sample preparation, immunostaining, and clearing because those steps are fundamental. How's your experience? How was your journey to get to a good quality level, a good and established protocol for 3D imaging?
Yeah, in that sense, I think we were lucky because the technology, again, tissue clearing is nothing new. It's a centuries-old technique, but has been now refined to a level over the last decade, I would say, that is actually quite nice and scalable, applicable to industrial settings. So we didn't reinvent the wheel here. We used techniques that are very robust and well validated through both academic partnerships and now even some industrial ones.
You've also had the chance to try some of our antibodies validated for 3D immunofluorescence. Can you comment on them? How do you find them?
Yeah, I mean, staining in 3D is challenging. Getting large molecules through thick tissues is even more challenging. So I think the trick here is to find the proper antibodies to stain in 3D. And I have to say that it's one of the most tedious parts of creating these workflows, not only validating antibodies, finding the correct ones. So the Miltenyi ones, the few that I had the chance of testing worked remarkably well. Not only stained the tissue in 3 dimensions without any observable gradients, but were also bright and robust. So it really helped us get to what is interesting instead of trying to debunk or optimize these protocols even further.
Yeah, very glad to hear that. Let's move on to the next step, imaging. Why is light chain microscopy the best or most recommended instrument for 3D imaging? And why the UltraMicroscope Blaze specifically? What are the advantages that you see in this system overall?
Yeah, so I think for the type of work we do, right, maybe we're a little bit less obsessed about resolution, more about throughput and robustness of the workflow. Of course, speed as well. So in that context, I think out of many of the systems that I've tried, both commercial and custom made, I have to say that by far the Blaze was, in my opinion, the best compromise in terms of speed, quality, and robustness of results. So we can really get from essentially hundreds of samples which back in the day would take us maybe a month or 2 months of scanning work, to a couple of days now, which is a pretty significant improvement.
It is an improvement, but it could also be very overwhelming, right? With the data size and the amount of data and images to be processed. How does your pipeline look like?
Yeah, so I think that's the tricky part with 3D imaging, is that, again, the histology part was really optimized now. The imaging is clearly getting better and better as we speak. But the analytical tools, despite many of them existing out there and being open source, I think still you need the really specialized personnel and experts to not only understand what the data even is about, but also handle these pipelines. So we have been working with these type of tools for now over a decade, optimizing not only the algorithms that we apply for speed, robustness, quality of the data we get. And I think it's an exciting time because these now suddenly everything feeds really nicely into each other. The whole pipeline is streamlined and you can run large studies fairly quickly.
You're also extensively using AI to handle, process, and analyze your data. What are the advantages of that, and where do you see this moving into the future?
Yeah, so I think AI is way more scalable than traditional segmentation methods. So we use this extensively in image analysis tasks. But now I think the exciting thing is that we also expand the capabilities of AI analysis to more predictive aspects of our data. And I definitely see this becoming a more standard way of analyzing these very large data sets in the near future.
Now, one question regarding your applications. You can do protein, you can do RNA. What does the future look like in that direction? Where do you see the field moving into?
Yeah, so that is definitely really exciting, right? Because protein works fairly well. Again, it's maybe difficult to optimize antibodies in 3D. But for mRNA, showcasing how powerful these techniques can be. And actually, again, mRNA is a much smaller molecule. So all the difficult things that we face with protein suddenly are much, much simpler using mRNA. I think also the exciting part of this is combining both protein and mRNA in the future. And there has been some examples published already showcasing this. But I really see this as something that will become more and more mainstream in the future with 3D imaging.
Right. And if there's one feature you could wish for, if it's a speck of the microscope or a step that will make sample preparation easier or something that helps you with the data analysis, what would that be?
I think automation, but that's maybe slightly biased, right, for our application. I think automation for sure, something that can help the user preset a very standardized set of settings for us to then feed into our pipeline and run analysis in a standard way. And also, of course, on the automation side of things, like how do you mount large amounts of samples simultaneously on your microscope reliably, that you can track what samples are, who is who, and essentially just have a higher throughput of imaging throughout.
We've also seen some examples here of combining 3D microscopy with other imaging techniques, for example, multiplexing. So on one way, getting the atlas, the full overview of your samples, and then which allows you to identify a region of interest and then focus on that one and go into the details. Mm-hmm. Is that something that you see your company moving into, or do you see these as being a beneficial workflow?
Yeah, absolutely. I mean, I think that, again, 3D has its own strengths and weaknesses. And so does 2D. I think for the 3D part, it's pretty obvious that you get a holistic view of the organ in an unbiased way, but with limited spatial resolution. The good thing is that you can use these 3D spatial approaches to precisely identify areas of interest that you want to investigate. And I think that the Maxima or 2D workflow really beautifully feeds into this, where you would use the 3D approach to maybe identify specific sites in the tissue you want to focus on, and then follow up with a more precise, focused 2D experiment. So,
thank you very much once again for your time.
Thanks.